In this PowerPoint, we're looking at homeostasis in the kidney. So homeostasis is the way of maintaining the constant environment within the body. It is usually under negative control or negative feedback, where we are trying to return the conditions back to the norm. And the response may be under hormonal control. So, for example, controlling blood glucose levels is due to insulin, or nervous control, where if your temperature changes, you may get goosebumps. That would be a reflex action that would be under nervous control. So the main parts involved in homeostasis are the receptors that are sensing the change. They will communicate with the control centre in the brain, which will bring about the corrective mechanism to bring it back to the norm. And why we need things to be normal or keep this stable environment is it helps provide the optimum conditions for enzyme actions, and also helps to avoid osmotic problems in the cells and body fluids. So the kidneys. Two main roles of the kidneys are, first of all, for the excretion of waste. For example, urea made from the deamination of the excess amino acids. So deamination is removing the amino group, the NH2 group. Or creatinine, which is a waste product that is made from the breakdown of creatine phosphate that is made in the muscles. So excretion of waste is one job. And then the other function is osmoregulation, where the kidneys play a role in controlling the water potential of the body fluids. The structure of the excretory system. So know your structure, know the kidneys, the ureter, the bladder and urethra. Those are the main basic parts. That the blood will enter the kidney from the aorta via the renal artery at high pressure, and then is filtered. Reabsorption occurs, and then it leaves the kidney in the renal vein and joins the vena cava, going back to the heart. Inside the kidneys, the main areas that we have, so we have the cortex, which is the outer dark region, which is found just under the fibrous capsule, and it contains the glomerulus, Bowman's capsule, proximal convoluted tubule, and distal convoluted tubule of the nephron. The medulla, the inner region that contains the pyramids that extend into the pelvis, and in the medulla region we have the loop of Henle and the collecting duct of the nephron. And then we have the pelvis, which is the large central cavity where the ureter joins the kidney. So in the nephron, the main thing that happens, first of all, is ultrafiltration. So blood enters the glomerulus at high hydrostatic pressure. And the pressure is high because the blood is coming from a short distance from the heart. The afferent arteriole, as you can see in the diagram, is wider than the efferent arteriole. So again, that increases the pressure. And the coiling of the capillaries in the glomerulus restricts the blood flow and increases pressure. This high hydrostatic pressure then will force small components of the blood, the glucose, water, amino acids, salts and urea, out of the glomerulus capillaries into the Bowman's capsule. Larger components such as protein or blood cells are too large to pass across. So the structure allows this filtration to happen. First of all, we have the squamous, the flattened endothelial cells of the capillary wall. So they have small pores in them that let some of the materials through. Then the basement membrane, which is found between the podocytes and the capillary wall, act as an effective filter, and that's what you write. The basement membrane acts as an effective filter, and it will prevent blood cells and large plasma proteins from leaving the blood. Then we have the podocytes, which are tiny finger-like projections that have gaps in between them that will let materials pass through and filter into the Bowman's capsule. So we then produce a substance known as the glomerular filtrate once it's passed through those three layers. So how does ultrafiltration occur? Why does it occur? So it's due to the differences in the water potential between the plasma in the glomerular capillaries and the filtrate in the Bowman's capsule. So if you remember our equation that we learned when we looked at osmosis in AS level, that the water potential of a cell is equal to pressure potential plus solute potential. So in the blood, the pressure potential, the hydrostatic pressure, is high and is much higher than the pressure potential of the filtrate in the nephron. The solute potential, so in the blood, the solute potential is greater as there are plasma proteins present in the blood, but there are none in the filtrate. But overall, the effect of the pressure potential is much greater. It outweighs the effect of the solute potential. That will then mean that the water potential of the blood plasma in the glomerulus is higher than the water potential of the filtrate in the Bowman's capsule. So the filtrate, the materials will move through the glomerulus, and there's the overall movement of the water down the water potential gradient from the blood into the Bowman's capsule. And it is mainly due to the fact that the pressure potential is so great that it is forcing those materials across, and it outweighs the effect that the solute potential would have, which would be trying to draw the blood or the filtrate back into the blood. Once ultrafiltration has occurred, then there is selective reabsorption of some of the molecules. So the main molecules that are selectively reabsorbed are your glucose, amino acids, and water, and ions as well. Most of this reabsorption occurs in the proximal convoluted tubule, or the PCT. So the glucose and amino acids are reabsorbed by facilitated diffusion and active transport. So those cells will contain lots of mitochondria for active transport. Water is reabsorbed by osmosis, and small plasma proteins are reabsorbed by pinocytosis. The lining of the proximal convoluted tubule is composed of a single layer of epithelial cells, which are adapted to carry out reabsorption in a number of ways. So these cells have microvilli to increase the surface area for reabsorption. They have proteins on the cell surface for selective reabsorption through facilitated diffusion and active transport. So that will be moving glucose and amino acids across. There's a high number of mitochondria, which will provide the ATP required for active transport to move glucose and amino acids across. Then there is the infolding of the membrane close to the capillaries, which you can see it near the bottom of the diagram, which also will increase the surface area. And the blood capillaries lie close to the cells of the proximal convoluted tubule. So all of those help reabsorption to occur. The rest of the nephron is involved in osmoregulation, so maintaining the water levels in the body. And this involves the loop of Henle and the collecting duct. So the loop of Henle's main function is to produce a negative water potential in the interstitial fluid of the medulla. How this happens is as follows. So first of all, the ascending limb will actively transport sodium and chloride ions into the medulla, making the interstitial fluid have a more negative water potential. So the surrounding the area surrounding the loop of Henle becomes more negative. As the fluid in the filtrate reaches the top of the ascending limb, it will have become more hypotonic or more dilute as ions have left the filtrate. So then when it enters the collecting duct, that will then allow more water to leave the collecting duct by osmosis into the medulla region, and that in turn will produce a hypertonic urine. The descending limb is permeable to water. So from the proximal convoluted tubule, it's entering into the descending limb. The negative water potential in the medulla that has been risen due to the ascending limb transporting those sodium and chloride ions out of it causes water to move out of the descending limb by osmosis into the medulla region. And it will then enter the blood capillaries of the vasa recta, which is the network of blood capillaries that surround the loop of Henle. This water has very little effect on the solute potential of the interstitial fluid. So because that water is getting taken into the blood and carried away, the surrounding area of the medulla is still staying very negative and has a very negative water potential. Now, as you move down the descending limb, the filtrate will become more negative because water is being lost, but ions are still present in it. So the water potential drops and becomes more negative and is said to be hypertonic as water is leaving the filtrate, but the ions remain. So be aware that in certain animals, particularly those that live in the desert, they will have a very long loop of Henle that will be allowing the movement of the ions out to cause more water to get reabsorbed from the collecting duct and also from the descending limb, but mostly from the collecting duct. And you may have to explain why that loop of Henle is so long. So this diagram's showing you just what we've gone through there. So you can see the step 1, where the sodium ions and chloride ions are actively transported into the medulla region, the interstitial region of the medulla. And if you look at the numbers on the ascending limb, you can see that the values are getting less, and that's because you're losing ions, and it's going to become a higher water potential as you move towards the top. Step 2, the descending limb. Water moves out by osmosis because the walls of the descending limb are permeable. So as it's losing water, the ions aren't leaving it, and you can see that the values increase as you move down the descending limb. So it's becoming more hypertonic near the bottom at step 3. And then our collecting duct, we have the water moving out into the interstitial fluid because the surrounding interstitial fluid has lots of sodium and chloride ions in it. There's a very negative water potential, and that is drawing the water out of the collecting duct and producing then a more concentrated urine. The distal convoluted tubule is after the loop of Henle, and its job is really to further regulate blood composition. So the ionic and pH composition of the blood surrounding the tubule is adjusted, and toxic substances like the creatinine is secreted into the filtrate from the blood at the distal convoluted tubule. ADH is the hormone that's involved, or antidiuretic hormone involved in osmoregulation. So nowhere it is made, so it's made in the hypothalamus. So it will monitor, the osmoreceptors in the hypothalamus will monitor the solute potential of the blood. Then the ADH is secreted from the pituitary gland in the brain, travel through the blood, and its effect will be to change the permeability of the collecting duct walls by either opening or closing more water channels or aquaporins. So the collecting duct is the site where water regulation occurs, and it is because of the effect of ADH changing the permeability. So whether there's less or more ADH will decide whether there's more or less aquaporins open. So if the blood is too concentrated, so you haven't drank water for a long time, you've been sweating and so forth, the solute potential of the blood becomes more negative. The osmoreceptors in the hypothalamus detect this negative water or solute potential in the blood, causing the pituitary gland to release more ADH into the blood. The walls of the collecting duct and the distal convoluted tubule will become more permeable. The aquaporins in the collecting duct open. More water will then get reabsorbed into the blood, and the blood then will have a less negative solute potential as it loses the water. The urine then becomes more concentrated or hypertonic and has a lower volume. The opposite will happen if the blood is too dilute, so you've drank a lot of liquid. So again, the solute potential is detected by the osmoreceptors, which will mean that less ADH gets released, so less aquaporins in the collecting duct open, and the permeability of the collecting duct is then reduced. Less water gets reabsorbed into the blood, and then more hypotonic urine, is produced. So that is the kidneys and their role in homeostasis.